So I will skip this slide. And I also know that now, after this beautiful talk, you know what is m6A. But just for you to remember, m6A is basically the addition of this methyl group to the N6 of the adenosine. And m6A can be written, but it can be also erased. And this confers this modification a great regulatory potential. m6A can be read by reader proteins, and the most known ones contains the domain YTH, which is able to bind the m6A. And m6A has been demonstrated to have a role in all the steps of RNA metabolism, including splicing, translation, but also, as you saw, stability, localization, and export. Usually m6A occurs in specific consensus motif. At the beginning of our research, nothing was known about the role of this modification on circRNAs. In order to carry out our study, we chose an example molecule, circRNA 609. It is an optimal example molecule for us because circRNA 609 was deeply characterized in our laboratory, and we previously demonstrated that it is involved in master cell proliferation. And also, it has another interesting feature. It comes from the second exon of its primary transcript, and it contains two start codons that upon circularization are in frame with a stop codon. And we demonstrated that they can be used to translate circRNA 609 in two different proteins. But first of all, we wanted to verify if m6A were contained into circRNA 609. An antibody against the m6A exists so that you can immunoprecipitate all the RNA containing m6A. And as you can see here, circRNA 609 is rich in the IP fraction, even more than its linear counterpart, but also of the positive control we used. And we used ATP50 as negative control. So in order to investigate the role, once established that the m6As are on circRNA 609, in order to investigate their role, we knock down MET3. MET3 is the main methyltransferase on the mRNA in the cell. And as you can see, the downregulation of MET3 is provoking a strong decrease of circRNA 609 RNA level. This is accompanied by an increase of its precursor level and its linear counterpart level. In order to extend our analysis, we performed an RNA-seq upon MET3 knockdown. Among the deregulated circRNAs, the most of them are downregulated, indicating that the knockdown of MET3 preferentially provoked downregulation of circRNA instead of upregulation. But we wanted to investigate the processes that were the cause of this downregulation. So we started looking for hints, analyzing the behaviors of these circRNAs linear counterparts. As you can see here, very few linear RNAs are concordant with our circRNAs, meaning that they are going up upon the MET3 knockdown when the circRNAs is going up, or vice versa. And so we excluded the transcription regulation or regulation due to indirect effect. The most of the circRNA are discordant, and they are not behaving as their circular counterpart, and in particular, about the 70% of them are not showing alteration at all. So we exclude some effect of competition between the circular and the linear production as cause of this downregulation. But we focused on circ biogenesis and circ decay. So from this analysis, we concluded that linear RNAs usually do not mirror the changes in the expression of their circular counterpart, and also that the MET3 mediated effect on circRNA is not the result of a transcriptional regulation, and neither of a competition effect. In order to exclude one of these hypotheses, we started from decay. We performed actinomycin treatment, and if you expect that your factor is acting on the decay, basically you expect that in the scrambled knockdown condition, the level of the circRNA in this case is going down with a different rate, since actinomycin stop transcription. So as you can see here, we have no difference between the scramble and the SI condition in the decay, in the rate of the decay of circRNA F609. So we concluded that no, MEC3 is not affecting the decay of circRNA F609, or at least it's not stabilizing it. So we focused on back splicing, and we focused on, in particular, YTH-DC1, which is a m6A reader known for its role in alternative splicing. And as you can see here, interestingly, we have a downregulation of the circRNA upon the knockdown of YTH-DC1. This is accompanied, yes, by the increase of the precursor level, but it's not accompanied by an increase of the linear RNAs. This suggested that the increase of the precursor is not contributing to the formation of the linear RNA. And this also suggests that there could be something on the precursor level that is distinguishing the precursor undergoing splicing from the one undergoing back splicing. And of course, it is not sequence. Maybe it could be m6A or other RNA modification. We confirmed that this effect is not cell type specific. Indeed, we carried out the knockdown of YTH-DC1 not only in HeLa cells, but other cell lines. And as you can see, we got the same results every time we performed our experiment. Also, in this case, we performed an actinomycin purchase experiment, and also in this case, we revealed that basically YTH-DC1 has nothing to do with the stabilization of the circRNA. Since while we were carrying out our experiments, a paper came out describing YTH-DC1 as an export factor for circRNAs. We also verified that there weren't problem of localization of the circRNA upon YTH-DC1 knockdown, and there isn't. We analyzed previously published data of YTH-DC1 RIP, and we found out that circRNAs are more rich than their linear counterpart in the IP fraction. Also, in this case, we wanted to extend our analysis, and so we performed an RNA-seq experiment. And also in this case, basically, we found out the same data. So upon the YTH-DC1 knockdown, the most of the circRNAs that are deregulated are downregulated. And also looking at the linear counterpart, we noticed that basically the most of them are discordant and they are behaving differently from their circular counterpart, indicating that it's not a problem of transcription and regulation, but it's some kind of other problem. Also in this case, we showed that there is no alteration for the vast majority of the linear counterparts. And so we concluded that also in this case, this splicing is affected. We wanted, again, to focus on circRNA F609 to validate our results. And so we analyzed m6A clip data in order to precisely localize the m6A on circRNA F609. This is the exon of circRNA F609, and every blue dash indicates an m6A. From this data, we got several information. First of all, there are many m6A on the exon 2 of the F609, so the exon undergoing circularization. Second, we performed the analysis both in the nucleoplasm and in the cytoplasm. And as you can see, there are m6A that are detectable in both the compartments, indicating that some m6A can work in the nucleoplasm where splicing occurs. Also, we performed this analysis in the poly-A plus fraction. And interestingly, here you can see that there are some m6A that are detectable in the total RNA, but you cannot detect it in the poly-A plus fraction, indicating that they could account for some RNAs without the poly-A tail, such as circRNAs. So we wanted to make mutants, of course, of this m6A, and so we took advantage of a vector able to overexpress circRNA F609 as a circular RNAs. But first of all, we wanted to verify the legitimacy of this tool, and so we checked the behavior of this circular RNA overexpressed in our cells and upon the knockdown of the factors acting on the endogenous circular RNA. As you can see here, the circular RNA produced from this vector is enriched in the m6AIP, so it contains m6As, and upon MET3 knockdown and YTH-DAC1 knockdown, it's behaving basically as the endogenous one. So we went through our experiment and we made like really many mutants as soon as we got the right one. I want you to focus on this mutant in particular, where we mutated the A alpha, beta, and 2. As you can see, we perfectly reproduced with this mutant the knockdown of YTH-DC1. Indeed, the mature product, the light gray one, is decreasing, while the precursor, in the bar colored in dark gray, is increasing. And also, it's interesting to notice that in this combination of m6A, we mutated the m6A alpha and beta that are detectable in the total fraction of the RNA in the m6A clip data, but not in the poly A plus fraction. So they could be specific for circular RNAs. Then, to validate our experiments, we tried to check the sensibility of YTH-DC1 of the mutant to YTH-DC1. And as you can see, this mutant is no more responsible with the downregulation of this factor, indicating that maybe we mutated the A's that are read by this factor. circRNA F609 can also be translated, so we next asked if the m6A abolishment could affect circRNA F609 translation. We got many effects on translation of the circular RNAs, but I want you to focus on these mutants. And as you can see here, the RNA is not impaired at all, but still we get a decrease, and a strong one, of the translation of the circular RNAs if compared to the wild type version of it. So our next question was, is this effect specific for our circular RNAs, or is just due to the sequence itself? So what if we express our circRNA in a linear conformation? So we construct a vector able to overexpress our circRNAs as a linear RNA, and it's solved as a linear orth with a cap and the poly A tail. As you can see, the pili in this case, I refer with pili to this transcript, it's enriched in the IP fraction, so it contains m6A. But when we mutated the A's, the most representative A's in this vector, we couldn't have any effects and any variation on the RNA level, but also in the protein level. So we concluded that the effect we saw with the abolishment of the m6A is due to the circular conformation of the sequence itself. We were then looking for the factors involved in this regulation, and we investigated these two factors, YTH-DF3 and the F4G2, because they are known to enhance cap-independent translation. As you can see here, the downregulation of YTH-DF3, but also the one IF4G2, is provoking a strong decrease of the protein level from circRNA F609. Coherently, when we overexpress these factors, then we get an overexpression also of our protein. When we try to downregulate these factors on the linear version and check the protein level from the linear version of circRNA 609, we don't have an effect. So also in this case, YTH-DF3 and the F4G2 specifically act on the circular RNA. in order to exclude indirect effects, then we performed the immunoprecipitation experiments of YTHDF3 and AF4G2. And as you can see here, YTHDF3AP is enriched of circRNA m609 as well as the one AF4G2. It's interesting to notice that YTHDF3AP is enriched also of the linear ZNF609, meaning the endogenous one, the mRNA coming from the locus of ZNF609. But you cannot say the same for the AF4G2IP. This is interesting because AF4G2IP is a factor involved specifically in CAP-independent translation, while LINZ and F609 is undergoing to a CAP-dependent one. So in conclusion, we demonstrated that m6A can promote circRNA expression thanks to a mechanism involving MET3 and YTHDC1, that the regulation of the circRNA is not due to transcriptional effects or competition effects, but the backsplicing is affected. Our data can suggest that there is a hallmark of the precursor RNA distinguishing the molecules undergoing splicing from the one undergoing backsplicing. And also focusing on circRNA F609, we demonstrated that it has m6As that can affect its RNA level and translation, and that in this mechanism of the translation, two factors are involved that are YTHDF3 and the AF4G2 directly bind to it. So basically, this is another repetition of our conclusion. So basically, we have the precursor molecule. It can be marked by m6A. YTHDC1 can mediate the backsplicing of fraction of circular RNAs. And once in the cytoplasm, translated circular RNAs can be helped in translation by these two factors. And I want to thank all my co-authors and all of you for your patience and obviously hope your attention. I have a good question. Carlos, I cannot hear you.
Thank you very much for your presentation. So someone have a question that or something that you would like to add, or you can write it on the chat. Okay, so yeah, I was wondering about this experiment that you did in which you look into the translation of the circle, but in there you have kind of mixed effects of the circle that it's already downregulated and the protein level. So I was wondering if you have found a way to kind of uncouple the methylation, like the production and the methylation, like you don't affect the production of the circle, but you still deposit, like you have still the mark on the circle to kind of see if there's really an effect on.
Okay, basically we started producing mutants for this reason, because we wanted to investigate the level of translation upon MET3 knockdown, but at the end we got like 50% of the RNA less than in the control condition, so obviously the analysis was impaired. I have to say that the densitometric analysis, they revealed that normalizing on the RNA and taking all the structure of the case, there is like 20% less of translation on the wild type. But still, I would refer to the mutants in this case to evaluate the effect on translation, because it's the only way to date for me to distinguish between the RNA, the MET3 effect on the RNA and the one on the proteins. Sorry.
Okay. I was also wondering if someone tried to do like a dominant negative of the reader that just binds but does not have the actual functions, and then you would block the cytoplasmic function of m6A. Like, okay. And the other thing that you mentioned at the end is the m6A peaks in the iClip data. So for this particular example, the peaks are in the 5' UTR, and the peaks are in the 5' UTR. So I'm wondering how often, like if you, in your mutants, did you look at where are these circles? Are they mostly coming from coding exon sequences, or they are coming mostly from 5' or 3' UTRs?
Okay, okay, I'll try to answer the second question, the first question, first the second. So because I'm here, yes, there could be a bias because you know the circular RNAs are enriched basically in the first exons of the transcript, and m6A is enriched in the exons as well. So there could be, but we performed actually a bioinformatic analysis revealing that wherever you take your circular RNAs, then it usually has more m6As than the other exon of the same transcript. So it looks like circRNA are like dense of m6A, let's say like that. While for the, but still there could be a bias. I mean, unfortunately, it's a coincidence, or maybe not. Maybe it's not a coincidence that exons that are enriched of m6A also produce circular RNAs. I don't know. Correlation doesn't mean causality, but yes, it could be. And then the other question was about, please remind me. Carlos?
No, I think my question was centered on this part of the modification in the 5' UTR, if it was a general feature or.
Okay. And that's it, basically. What else on this part? No, I think that's all. I mean, there is much more to study in this field because circRNAs are so studied now, but still there is much more to understand also in the m6A field. So to date, my only answer could be this one.
So if there's not any more questions, then I guess we are close. We're going to close this symposium. So thank you very much, everyone, for joining us. It was a great experience because we had a lot of people that is working in basically the same, like in similar topics surrounding the circular RNAs. We had a great opportunity to really see this from different points of view. And Mariela, I don't know if you also would like to add something or.
Yeah, I mean, I would like to thank the organizers for inviting us, and I hope we will see more in the future, more RNA-related mini symposiums or talks in this project. And I just invite you to subscribe for the project if you are interested in the topic, and see you next time.
Thank you very much, all the speakers and the moderators for this symposium. I think it was a really great symposium with a lot of new data on circular RNA. And we invite you to follow our initiative and hope to anyone interested in hosting or organizing a symposium, just contact us and we will help you through the way. Thank you very much to anyone.